and now moving on to part 2, looking at chapter 3. And here we are focusing on this section of the carbon cycle, looking at the hypothesized production of detritus by sponges, which would make it available to detritivores. To start, detritus is a mixture of non-living organic matter derived from living sources. So things like shed cells, algae, broken down fragments of algae, feces, etc. And to introduce another important term, it accumulates in the turf algae that covers dead coral surfaces, and this forms the epilithic algal matrix, or EAM, which is an important location and resource for detritivores to graze. And a fundamental question is, do sponges even produce detritus? Now, the sponge loop hypothesis was originally based on studies of this species, Halisarca caerulea. This is a cryptic sponge that grows in very, very thin layers, and it has a really high cellular turnover, so it sheds its collar cells very fast. And this is the detritus that it supposedly produces. But for those species that live above the reef framework that emerge from the reef, we're not so sure whether they make detritus. Some studies have concluded no, they don't, and the study used the same kind of index sampling that we did in chapter 1, only they also measured particulates. Another recent study filmed the in-current ostia of some of these emergent species and found that there was these particulates and mucus coming out of these ostia, which they considered to be a kind of detritus production mechanism. And here's a video from that sneezing paper, which they called it sneezing. This is an Aplycina from Curacao, and you can see the particulates coming out of the ostia kind of clumping together, and then eventually the sponge contracts and then they get released onto the reef. So it's a pretty interesting video of a process we haven't really observed before. So could emergent sponges be producing detritus? Maybe, I don't know. The video is certainly compelling, and I'm interested to see what else they find out about these particulates, especially whether they even are detritus. But I'm not entirely convinced yet. And truthfully, I'm not too concerned about this question because I care more about the big picture, the function of the sponges in their environment. So for the sake of argument, let's be generous and assume that sponges do produce detritus. What I want to know is, do they produce a lot of detritus? More specifically, do they produce so much that if I go out and grab a handful of detritus from anywhere on the coral reef, that I can find a sponge signature in it? This is a relevant question because according to the sponge loop hypothesis, the estimated amount of carbon transferred back to the reef by sponges, shown here, is an order of magnitude greater than that transferred back through the microbial loop, and roughly equivalent to the particulate organic carbon production by primary producers, of which much of this gets trapped in the EAM as detritus. So if the sponge loop is this major of a process, then we should be able to find a huge sponge signature in the detritus. So our objectives for this chapter were to look for that sponge signature and to determine the contribution of sponges to detritus relative to other detritus producers. To do this, we set up a sampling experiment. Again, this was at Caribou Key. And we collected samples of detritus from the EAM, as well as stuff that we captured in trays that we deployed under reef overhangs. And this was a very generous test of the sponge loop hypothesis to capture any outfall of shed cells from things like Halisarca that grow in the nooks and crannies on the reef. And then we also collected samples of potential sources of detritus, so things like algae, benthic cyanobacterial mats, fish feces, and also the tissues of 3 cryptic and 3 emergent sponge species to get that full representative sample of the sponge fauna. And we also deployed sediment traps to capture stuff that was falling out of the water column and be able to separate that from the detritus that is produced on the benthos. And we repeated this sampling in July 2018 and March 2019 and analyzed these samples using stable isotope analyses. So here's an isotope biplot, and each point shows the average delta value of the detritus samples and possible sources that we collected in July 2018. So here we can see the EAM detritus in the middle, and then all of the potential sources of detritus around it. And in general, we saw some grouping of species from the same detritus categories. So the algae and cyanobacteria were down here, and then the sponges were up here higher on the nitrogen axis. Also, the herbivore feces grouped out with the algae. So this is the blue tang and surgeonfish, while the spongivore feces, this one angelfish sample, is grouped out with the sponge. And this made sense to us since the isotope ratios of these fish feces should match the main components of their diet. Next, I used a stable isotope mixing model to determine the composition of detritus based on the isotope values of the potential sources. So what this is showing is that for our 2018 samples, the algae, BCM, herbivore feces, and sediment trap samples were the main contributors to the detritus. And to note, I did not choose to group the sample types this way, but these 4 groups were highly correlated, and so I had to group them to make the most reliable mixing model estimates. And now here's the biplot from the March 2019 samples. And this is showing a few key differences. So for one, there was this interesting shift in the tissues of these 2 sponge species, Aplycina and Chondrilla. And these guys had an increase in carbon delta value and a slight decrease in nitrogen delta value. And overall, things were just more clumped together and had larger error bars. So we have more tighter clustering of more variable sample types. And because the isotope values were so much different and a little more variable, this really changed the mixing model results. So here we see nothing really stands out as a main contributor as to the detritus, either to the EAM or the detritus that we collected in March 2019. At the same time that we collected samples for stable isotopes, we also collected them for DNA metabar coding to see what kinds of genetic material were in the detritus. And to run these samples, we collaborated with the Center for Environmental Genomics Applications, or CEGA. So we sent our samples to them up in Newfoundland, and they analyzed them using their proprietary method. To briefly summarize what they did, first they optimized the DNA extraction methods for each sample type, and then they amplified these 3 markers to generate 3 amplicons per sample. And then these markers were chosen based on our target groups, so the metazoans, algae, and cyanobacteria. Then they sequenced the samples and cleaned up the data to produce this list of exact sequence variants, or ESVs, that they then screened against the NCBI database to look for matches and assign taxonomy. At a glance, they assigned taxonomy to a little over a third, or about 295,000 ESVs, and they detected 56 phyla in the detritus, including 15 metazoan phyla. So this summer, when I really started looking at these data, I realized how neat it was and how much information we had here. But at times it was very overwhelming, and I thought at times I was going a little crazy trying to analyze all these data. But at that time, it helped to step back and really keep the main objective in mind and step back and look at the data as a whole based on these major organism groups. So that's what I'm going to do. So starting with metazoans, here are the top 10 metazoan ESVs that we detected in the EAM detritus in July 2018 and March 2019. And these percentages show the frequency of occurrence, basically the percentage of samples that each taxon was detected in. This was our way of kind of getting around the problem with this analysis that it'll tell you if it found a species in a sample, but it doesn't tell you how much of a species it found in a sample. And this frequency of occurrence metric can approximate abundance as long as you have multiple samples. And this is in order of decreasing frequency. So at the top, we have some copepods and other arthropods, and some things like acelians and nematodes. And these might be mostly benthic taxa that live in the EAM. And if we keep going down the list, we see some other stuff like cnidarians, platyhelminthes, annelids, even chordates, and things do start thinning down near the bottom here. And now I don't include sponges in this table because we wanted to make a whole separate table just for sponges, but keep some of these percentages in mind. And now here are a table of sponge taxa, and we found 36 taxa across all of our different sample types, but only 12 in the EAM, and none of these were in more than 50% of samples. So sponges fall at the lower end of the spectrum of frequency, even just among metazoans. And for these 6 sponge species whose tissues that we collected, we chose these species because they are both abundant on the reef and because these 3 cryptic species were really the foundation of the sponge loop hypothesis. And we did verify the species ID of the tissue samples that we collected using genetic sequencing. But in the detritus samples, we only found chondrilla in 1 sample of trait detritus, and we didn't find any of these species in any of the EAM detritus samples. So it's pretty notable that even this Halisarca was absent. This is that exemplary detritus producer. So if it is rapidly shedding its cells and turning them over, which would presumably leave a genetic trace, we didn't find a signature of it. And before I move on from metazoans, there's one more thing I wanted to point out that stood out to me. This was this relative abundance of cnidarians, more specifically corals, but not scleractinian hard corals, like many previous studies have reported to be important sources of detritus. We actually didn't find any scleractinians in the detritus, but we did find a lot of these octocorals, so things like gorgonians. And indeed, the fore reef at Caribou is just full of these arborescent octocorals, and as well as sponges. And in my literature search, I didn't find much of a mention of how octocorals might be contributing to detritus. I actually was surprised at how little information there is about octocorals relative to other benthic groups, but I did include some citations to 3 nice recent reviews and studies that all mention the rise of these octocoral forests, something like we could almost be seeing here on the fore reef at Caribou. So it's pretty crucial to learn more about this group, its functional roles, and also how it's contributing to the detrital pathway. Now, any abundance of metazoans was really just eclipsed by primary producers, which for one agrees with our stable isotope results, at least from July 2018. And also, this wasn't really surprising since algae are generally considered to be major sources of detritus. And the taxa in bold here are the matches to the tissue samples that we collected. So we have, in our 2018 samples, Lobophora was in 80%, and Morea producens was in 40%. And I also included some, so these are pretty high numbers for such specific IDs, but I included some higher order taxonomies for those groups as well, just to show how frequent some of this stuff was. And I also included things like coralline algae and sargassum, just that stood out to me because I recognize them. But this is a very cherry-picked table, just full disclosure, because when I look at the top 50 primary producers, we see a much lower diversity. So we have some things like rhodophyta and cyanobacteria, but this was really dominated by the Bacillariophyta, which are the diatoms. Also, it was notable that 49 out of the 50 of these taxa were in 100% of the July 2018 samples, but there was a sharp decline in the March 2019 samples as we go down the list. And in fact, there were thousands more ESVs in the July 2018 detritus. So there's clearly some seasonal differences that drove the big changes that we saw. For one, we personally observed big differences in the overall energy of the system between July and March. So in July, we had these very calm, hot conditions, but in March, we had very turbulent, surgy, and windy conditions. We actually lost several field days due to weather in March. Also, we know from previous studies that the irradiance and water clarity are much higher in the summer months at Caribou. So in both July and March, the wind and water predominantly moves from offshore to onshore. So there could have been more settlement of oceanic stuff like diatoms, especially in July when it was calmer and gave these things time to fall out of the water column or gave them time to get eaten by that wall of mouths on the reef. Also, the higher irradiance could have caused more primary productivity, which would have led to the higher frequency of primary producers that we saw, and especially if some of this stuff might have been blooming, which I've seen some reports of at Caribou. And the higher irradiance in the summer would also help explain the differences in the tissue samples from these sponge species that we saw, Aplastina and Chondrilla. So these are sponge species that contain photosymbionts, and those preferentially fix lighter carbon. And so if they are more active in the summer months when there's peak irradiance, this would likely have been reflected in the tissues of those sponge species. So now to summarize and put things in perspective, regardless of season, it seems like there are various sources of detritus, but sponges do not really appear to be anywhere near the top. In fact, they seem to be pretty minor contributors, especially compared to other things like algae. And just to recall this figure, our data are not really consistent with these estimates that there's more detritus from sponges than from primary producers or other things on the reef. It doesn't really add up, at least on Caribou. Also, our observation of potentially high oceanic inputs to the detritus are consistent with previous studies of Indo-Pacific reefs that also found large oceanic inputs to the detritus. And then when there's high flow over the reef, the stuff gets pushed back downstream to back reef habitats. And then detritivores graze this stuff and move it up the food chain. Now, unlike Indo-Pacific reefs, there's a lower abundance and diversity of detritivores in the Caribbean. And so if 4 reefs on the Caribbean do receive any surplus of detritus, for example, from groups like algae or octocorals or cyanobacteria, these things that are becoming more abundant on the reef, these surpluses could supplement the natural sources from the ocean and then potentially lead to there being more detritus that can be grazed by the detritivores. So this stuff could end up building up on the reef with unknown consequences. So that concludes the work that we did at Caribou Key, looking at both the DOM and the detritus. And now moving on to another part of the Caribbean for this last part.